Thanks to the organizers, and I'm pleased to be here in person again. And today I apologize for a rather generic abstract I sent in, but what I'm going to talk about today is some structure and functional studies we've done recently on the ochratoxin A binding aptamer. Is that me advancing or you? Okay. So just as an introduction, in my lab, we study how aptamers work. We have a focus on small molecule binding DNA aptamers. We don't do any selection. We don't really try and develop biosensors. We're doing structure function studies, aptamer, small molecule complexes. And we do things like we use isothermal titration calorimetry to measure the affinity and thermodynamics of ligand binding. We use differential scanning calorimetry to study the stability and folding thermodynamics of aptamers with and without their ligands. And our structural studies are based on NMR spectroscopy techniques. And I can't point, but if you see me in previous talks here in the aptamers meeting, I've really worked on the cocaine binding aptamer a lot, but today I'm going to work on something different, and that's the ochratoxin A binding aptamer. So it's interesting that Gregory Penner is speaking later today, but he originally selected the ochratoxin A binding aptamer that we're working on, and as you can see, it's predicted. Well, first of all, there's the ligand. I might have a pointer. Oh, I got a pointer. Very good. So here's the ligand. It's, I like to think of it as a derivative of phenylalanine. So here's your, N-C-alpha carbonyl in the phenyl group. The aptamer is, that's predicted by our collaborators, Richard Mandeville at Guelph. It was predicted to contain one of these duplex G quadruplex type structures where you have a helix leading into two G tetrads. So the G quadruplex is made up of two G tetrads with the loops arranged like this. Richard's lab worked out, I won't mention how he did it, but the paper down here outlines how he worked out the topology of the aptamer. So as I said, first thing so Richard, was talking to me where it's a university close by, and it encouraged me to work on the aptamer. And the first thing we did was to double check that it bound. And so we use ITC, and this is the only ITC I'm going to show, but yes, the aptamer does bind. It's slightly complicated because the aptamer needs to be dissolved in DMSO, and DMSO has a large nonlinear heat of dilution. But we figured out conditions where we get a nice repeatable, quite tight binding at 57 nanomolar, and it's quite exothermic, so it's relatively easy to detect by ITC. So we use, as I said, NMR spectroscopy to look at the structure of the aptamer, and I'm not going to show too much NMR. We look at are the iminoprotons in base pairs. So whether you have an AT or a GC base pair, there's a highlighted in orange here, an iminoproton that's protected from exchange with the solvent and shows up in the downfield region of your NMR where only aminoproteins occur. And in the case of the ochratoxin binding aptamer, here's a titration of the ligand, adding in ochratoxin. On the bottom, we have the free aptamer, and up on the top, we have the ligand-bound aptamer. And what we see down here in the bottom in the free aptamer is an example of a really ugly NMR spectrum. Lines are broad, not all the expected peaks are there. But as we add ligand, and we see we only have to add a small amount of ligand, the signals sharpen up, the expected number of peaks are all there. And, based on my years of work with the cocaine binding aptamer, what we thought happens is, we've, have some sort of structure switching event going on. where we have an unfolded aptamer in the free state, lines are broad. So in the free state up here, ligand binds, and we get the structure forming in the presence of ligand. So this is, it's interesting that this is similar NMR spectra that we've seen previously for the cocaine binding aptamer, at least a short stem 1 version, which is unfolded in the free state, folds in the bound state. So, we worked also with a collaborator of mine, Cameron Macarath at Bordeaux, and through using a series of C13, I must say we cheated. For any NMR person out there, we can say we cheated, and basically made a lot of samples that are C13, N15 labeled at the G position, as well as some of the other positions in order to get the assignments of the NMR. And so we've specifically assigned all the peaks in the NMR spectrum of the bound ochratoxin aptamer, ochratoxin A binding aptamer. The amino assignments are shown here. There's an exceptionally unusual peak that lies upfield from here. And what this did is at least confirm the topology of the 2G tetras and confirm that we have at least 3 base pairs here in the tail. Now, I'll point out that we had kind of unusual conditions. The aptamer was selected in the presence of 20 millimolar calcium chloride. And when I first started working on the project, as an NMR person, we didn't want to work with calcium because we like to use a phosphate buffer because a phosphate doesn't have any protons in it. And if you have phosphate and calcium, you get calcium phosphate precipitating. We ended up working in deuterated tris because, as it turned out, calcium is essential for binding. And that's, that's shown in this slide here. So in this slide, we're looking at the NMR spectrum, the downfield aminoprotons of the calcium-free, ochratoxin-free aptamer, and essentially the spectrum again looks ugly. Not many peaks, what's there is broad. We add in ochratoxin, and we see this peak up the middle. That's as a result of the ochratoxin. It's the kind of like the amide on the phenylalanine showing up. So we see the ochratoxin A coming in, but we don't see a nice improvement in the NMR quality. So, and we see little to no change in the NMR spectrum, indicating we don't get binding the ochratoxin without calcium, which was, confirms, the selection was done with calcium. It's not surprising that it needs calcium for binding. We then took this sample, which is shown at the bottom here, added in calcium. Essentially what I'll say is the spectrum goes from looking ugly as in the previous slide. The peak due to ochratoxin shifts over, and as we add in more and more calcium, the spectrum improves, and we get the same spectrum as we got shown previously. If we monitor this, this addition of calcium with ITC, the affinity of whatever observed interaction we see, which I think is a mixture of ochratoxin A binding and calcium binding, has an affinity in the 100 micromolar. So next thing we did was use differential scanning calorimetry to look at the stability of the ochratoxin A binding aptamer in the presence of ochratoxin and in the absence of ochratoxin. And DSC measures the stability. The peak height here indicates the melt temperature of your, in this case, it is a DNA aptamer, so the melt temperature of your DNA. And we see a couple of things. We see the melt temperature of the ochratoxin A bound aptamer increasing compared to this is the in the presence of calcium. So the calcium bound aptamer is shown here, melt temperature increasing. The area under the curve represents the enthalpy of binding, and the enthalpy, well, the difference in the area under the curve represents the enthalpy of binding. So the total enthalpy of unfolding is the area under the curve, and it increases in the bound aptamer because you have the enthalpy of binding present as well. So this looked great. And then when, thought about it for a minute, and we sort of thought that, if the free aptamer is unfolded, we shouldn't really have any observed signal. We shouldn't have a peak here for the unbound aptamer if it's unfolding. There's nothing to unfold. We shouldn't be measuring an unfolding event. So what we thought was that the calcium plays a role in stabilizing the aptamer structure. So what we did next was do DSC without the binding buffer with the calcium. And in fact, we added some EDTA to chelate any calcium that may be present. And in this case, we're looking at the unbound aptamer in the presence of calcium and the ochratoxin A unbound aptamer, as well as a calcium-free aptamer. And we really do get a flat line, indicate that there's no folding in the absence of calcium, which brings us to the fact that within the presence of calcium, we do see an unfolding signal in the DSC. And what we think is going on in what we can do to explain the ugly, for want of a better looking term, the ugly NMR spectrum of the calcium bound but ochratoxin free aptamer is that we have G register dynamics present. So I know this is small, but we have our 8 Gs involved in 2 tetrads. And in the sequence adjacent to the 8 Gs are other Gs. And what we think is going on, if you could advance the slide, please, is, I'd have to put on my glasses, I can see it here. We could have a G slip down, we can have another arrangement, we still have the 2 G quadruplexes present, but one of the strands has slipped down, the one in the middle there that I can't read without my glasses, G13, I believe. Or another strand could change position. We would still form the 2 G quadruplexes, or the 2 G tetrads in the quadruplex, but we have a different arrangement of Gs in there. And there's another one, the other strand can be displaced. And, et cetera, et cetera. You can imagine, whoops. You can imagine a whole bunch of slippage events taking place. And that's what we think is going on in the free NMR, and that we have a whole series of different registers the Gs are in. So it's really a dynamic equilibrium between a whole bunch of different, all contain 2 tetrads, but they contain different arrangements of the 2 tetrads. And then in the presence of ligand, advance one more, please. There we go. We bind to the ligand, we bind the aptamer to the ligand, locks in the one conformation that binds, and we get a whole sharpening of the signal, and we get a really beautiful looking NMR spectrum in the presence of ochratoxin A, and we don't have any of the other dynamics with the other possible quadruplex structures. So I think that was, there we go. I didn't actually press the button, so you had me fooled that I was doing the changing all along. Yeah, so I just want to end with a little preview of the structure of the aptamer we have almost finished. I will emphasize almost finished, and what we have here is a preliminary structure of the ochratoxin A binding aptamer in the presence of calcium bound to ochratoxin A. In collaboration with my colleague Cameron in Bordeaux, what we've shown is that we do have 2 G quadruplexes, just as predicted by Richard Mandeville's CD studies. But what we also have in the structure is, I think, really, really cool, is that we have 2 base triples on either side of the G quadruplex. that looks better. So we have two base triples, either side of the G quadruplex, and we have a base pair underneath the base triple on what we call the bottom of the structure, or the non-ligand binding side of the structure. So the ochratoxin binding site is up here. The ochratoxin actually doesn't make contact with the top quadruplex. It makes contact with this base triple. The calcium binding site, we've done some cool experiments where we replace calcium with cobalt hexamine, and we've looked at NOEs from the cobalt hexamine to methyl groups, and we see NOEs to both T30, T3, as well as the methyl group on the ochratoxin binding up here. And on the right are the current structures we have. Now, the structure in the structures are calculated in the presence of the ligand. I'm not showing the ligand here, but on the right in purple, or sort of a row, let's call it not purple. Magenta, I think the color is. In magenta here is the ochratoxin, and it really fits in nicely right where the junction between the short helix meets the top base triple. And I'll just mention, besides the fact to emphasize it's a preliminary structure, there's a whole class of these duplex, quadruplex structures coming out now, and the binding site for ligand is always in the 3 or 4 examples that exist, where the duplex, or sometimes a triplex, meets the quadruplex, or at least in this case, the top base triple. So I just want to finish by thanking the people who did the work. Zach Churcher, who has a flash talk from back in Canada. He's graduating, having his defense next week, so hopefully graduating soon. Megan has done the DSC. She's giving a talk and has a poster. Aaron and Sujana have done some of the other work, and I'll just mention Emily and Rahith, who all have posters and flash talks here at the conference. And thank you for your attention. I'll take any questions.